Hello, I'm Julian Somers. I'm a part of the Baustein lab, and what we study is essentially mostly to do with LNC-RNAs, which I'll talk to you more about later on. But the main thing that we need to start with is the fact that we're studying the salt responses in the plant family Brassicaceae. And what we're looking at specifically in there is the effect of long non-coding RNAs and what role they play. So, salt. All of us know salt, I'm pretty sure. A lot of you have probably enjoyed very salty fries, even though you know that's probably not the best thing for you to eat. As a human, having too much salt can cause a lot of adverse health effects. You can have heart failure, you can have high blood pressure. I believe even kidney damage is up there. And it probably shouldn't surprise you that plants also can suffer from having too much salt. This is not from our experiments, it's something that I found online. I have the link right there. But you can see as you increase the concentration of salt that plants are receiving, they tend to shrink and not do as well. They tend to turn pale yellow and too much, and they will certainly die. So, why is this a big issue? The reason is because soil salination reduces crop yield significantly. As you can see, those plants become very small and not as healthy, and so they can't produce as much. And if you look at this, you can see that there's actually quite a lot of area that has very poor conditions as far as soil salination goes. Not very far from home, you actually have some very poor soil, which makes it very difficult to grow crops. So, how can we make plants grow better in these saline conditions? Well, one thing we're figuring out is that LNC-RNAs may be a possible answer to help with this. Long non-coding RNAs, called LNC-RNAs for short, have several functions. They are greater than 200 nucleotide long transcripts, and they don't code for a protein. That's why they're non-coding. And a few of their known functions are signals, decoys, guides, and scaffolds. As you can see by this cute little cartoon, they can do things such as bind to several proteins and act on chromatin structure in order to either prevent gene activation or allow it to happen. And essentially, all of these add up to they play a large part in regulating other genes. So the way that we classify LNC-RNAs is based on genomic location. What we're most interested in are these intergenic LNC-RNAs. These LNC-RNAs are in between protein coding regions, and they tend to be rather conserved, which means they are more likely to be important. And one reason that we love these intergenic LNC-RNAs for research is because since they're not overlapping with a protein, if what we do is interrupt their function by messing up the intergenic LNC-RNA, for example, making a tDNA mutant, putting transfer DNA into that sequence in order to mess up the order and make it not function and not be produced. If we mess up the LNC-RNA, we're not messing up any proteins. And so any change we see is because of the LNC-RNA. So of course, we're talking about salt. How do we deal with salt? Well, we're looking for conserved salt-responsive LNC-RNAs. And so that means LNC-RNAs that are found, the same sequence is found along many different species within the plant family Brassicaceae. And so you can see here a phylogeny tree of Brassicaceae. And you can see that it has the percent of homologous LNC-RNAs identified compared to Arabidopsis thaliana, which is a wonderful model organism. And you can see that as you get further away, you get fewer. And you can see here, we're looking at this more accurately than that, because we want to see which LNC-RNAs go further back and are the most conserved. So we want to know the ones that even all the way down here still have matches. And so we got 25 of those based on RNA sequencing data that was generated by another group also dealing with similar experiments. And so from those 25, we looked for homozygous tDNA mutants in Arabidopsis thaliana that were for tDNA insertions, so that we could look at those plants, see what is disrupted, see if they fare worse than the wild type. And so after gaining our, I believe it was 12 lines that we got, we started gathering data using root length assays. So what we were doing here is first we would prepare plates and we would put seeds on them. And they would be the starting plates. They would be put in the cold room for stratification, 3 days. We would take them out and allow them to germinate. And then we would transplant them to a new plate. And half of these plates would be salt, half would be no salt. We were starting out only using 75 millimolar salt on the salt plates. And we would mark the roots each day for 8 days, and after 8 days we would scan the roots, and then on a program online, on a computer, we would trace the roots, it would calculate the length for us, and then we would be able to produce graphs from that. You can see what it looks like here, transplanted, and then you see marking each day at the root. So this is what we expect to see. This is a simulated fake one of what we expect to see. And so you see that these two here are in the no salt condition. We expect that the mutants and the wild type should do approximately the same when there's no salt, because these are salt-expressive LNC-RNAs. And then we expect that when salt is present, the mutants should fare much worse. And so we got results for 12 mutant lines, and we found 4 that were very good candidates. So a closer look, you can see that the 3 stars means that there's a very significant difference between the wild type and the mutant when there is salt present. And this is only 75 millimolar salt. It's, by comparison, they can survive, we found that they're able to survive in up to approximately 125 millimolar salt. And so we're already seeing a pretty solid difference on some of these plants. And so what we're trying to do in general is identify the function of LNC-RNAs in the salt response of Arabidopsis thaliana first, and then we will broaden our approach to look at more of the Brassicaceae family. We have identified 4 strong candidates, and moving forward, we're gathering more data so that we can produce more convincing results. And we're also trying to learn more about the way that the LNC-RNAs work. We're trying slight modifications in our experiments. For example, we're attempting to use different concentrations of salt. Instead of just 75 millimolar, we've increased to also using the 125. We tried 150 as well. They kind of survived there, not the best. And then also, we're going to be starting up some more experiments that have to do with other species such as Camelina, sativa. That one seems to have a higher tolerance for salt, and so we'll be able to stretch out the difference more clearly. Acknowledgements. Thank you to all my lab, and your, and funding. Funding is great. Thank you.